Welcome to the deep dive. Today we're really digging into the bedrock of safety in labs and healthcare. The rules, the standards that keep things safe. Our mission for you today, simple. We want to quickly pull out the absolute must-know info from all those safety manuals, turning dense text into, well, practical knowledge.
And this is so critical. I mean, the second you walk into a place working with biological agents, bacteria, viruses, fungi, even toxins, you're facing a potential occupational hazard. It's inherent. Every single guideline we're going to talk about, really, from just washing your hands to, complex high containment labs, they exist because historically things went wrong. People got infected, stuff got contaminated, or worse.
Okay, let's unpack this. Let's start with the organizations that built this foundation, the big players who define safety.
Right. So here in the U.S., you've got OSHA, that's the Occupational Safety and Health Administration. They kicked off back in 1971. Their mission is pretty straightforward and make sure workplaces are safe for everyone. And honestly, they've had a massive impact. Since they started, workplace deaths have dropped by over 60%.
Wow, 60%. That's huge. Was there like one single policy that made such a big difference so quickly?
Well, it wasn't just one silver bullet, it was a combination. Things like mandating and engineering controls actually changing the physical workspace, and crucially, the right to know laws. That meant facilities had to, properly label chemicals, train workers about the hazards they face. That was big.
Okay, and then there's the CDC.
Exactly. The Centers for Disease Control and Prevention. They started even earlier, 1946. Originally, believe it or not, their focus was malaria control. Now, of course, they're the lead U.S. agency for, well, pretty much everything public health, from E. coli outbreaks to pandemics, vaccines, you name it.
And rounding out the big three globally is the WHO, the World Health Organization.
Yeah, WHO. Established 1948, they're the ones coordinating public health on an international scale, monitoring global threats like SARS or AIDS. And they were behind one of the absolute biggest wins in public health history, wiping out smallpox globally. And maybe less known, they actually reversed a pretty controversial policy on DDT spraying for malaria. They decided, after review, that the benefits in fighting malaria actually outweighed the environmental risks in certain contexts.
So these groups set the standards, but things can still go wrong. The source material mentions that really scary 2014 incident with anthrax at the CDC itself.
Oh, yeah. That was a serious wake-up call. They were trying to chemically sterilize Bacillus anthracis samples. They got initial negative test results, trusted them, and sent the samples out to other labs.
Right.
And about 6 days later, live anthrax colonies started growing on the culture plates in the receiving labs. Personnel had been exposed to live, deadly bacteria.
That's terrifying.
Absolutely. And the lesson wasn't just be careful. It showed a systemic vulnerability. Relying only on initial chemical kill results without, robust secondary biological checks to confirm no survivors, that protocol had a dangerous blind spot.
It really underscores why even the most basic controls are so critical. Before we get into the high-tech stuff, let's talk about the absolute cornerstone, hand hygiene.
Yes. And we have to mention the history here. Back in the mid-1800s, child bed fever, that's puerperal sepsis, caused by Streptococcus pyogenes, it was devastating. Mortality rates were hitting 25% in some hospitals. Then Dr. Ignaz Semmelweis noticed something. Medical students were going from autopsy rooms straight to the delivery ward without washing their hands. He suspected they were carrying, putrid matter.
Oh.
Right. So he made them start washing their hands with a chlorine solution between activities, and the death rate just plummeted almost immediately down to less than 1%. Dr. Oliver Wendell Holmes in the U.S. had similar findings around the same time.
That history really drives home why the CDC today hammers on that 20-second rule. Vigorous washing, soap, warm running water, getting under the nails, backs of hands, and using a paper towel to turn off the faucet, right? To avoid recontaminating your clean hands.
Precisely. It seems simple, but it's probably the single most effective thing we can do. It closes that contamination loop.
Okay, so now let's get into the main framework for lab safety. The biosafety levels. BSL-1 through BSL-4. These levels are linked to the NIH's risk groups, RG1 to 4, and they basically scale up the protection needed based on how dangerous the agent is.
Exactly. So BSL-1 or risk group 1, that's your starting point. Minimal hazard. We're talking agents like non-pathogenic E. coli, Bacillus subtilis, things that generally don't cause disease in healthy adults.
So where would you find a BSL-1 lab?
Think municipal water testing labs, maybe some high school biology labs, basic stuff. The requirements are standard lab practices, no eating, drinking, smoking, definitely using mechanical pipettes, not mouth pipetting anymore. And you need a sink for hand washing, basic hazard signs. Pretty straightforward.
Okay, moving up. BSL-2, risk group 2. Now we're dealing with agents linked to human diseases, but ones that are usually treatable. Interventions are available. So things like salmonella, strep throat, streptococcus pyogenes, hepatitis B and C, even HIV fall into this category.
Right. And the requirements get stricter here. Personnel needs specific training on handling pathogens. Access to the lab is limited when work is actually happening. Immunizations are often recommended or even required. OSHA, for instance, mandates the hepatitis B vaccine for healthcare workers who might be exposed to blood. And you have to be extremely careful with sharps, disposal needles, scalpels.
Here's where it gets really interesting. Let's look at the highest containment levels, BSL-3 and 4.
Okay, BSL-3, risk group 3. Now we're talking serious business. These agents are indigenous or maybe exotic, and they can cause serious, potentially lethal diseases if inhaled. Think Mycobacterium tuberculosis, the agent causing TB, Bacillus anthracis, anthrax again, yellow fever virus. Interventions might exist, but they aren't always effective or available.
So the lab itself must be quite different.
Oh, absolutely. You need supervision by scientists specifically trained for this level. Access is highly controlled, often electronic key cards, logs. No minors allowed, definitely restrictions for people who might be immunocompromised. Required immunizations or testing, like TB tests, are common. And critically, procedures that could create aerosols must be done inside a biological safety cabinet, or BSC.
What about the room itself?
Big changes there. The lab often has special ventilation, negative air pressure, so air flows in, not out. Exhaust air is filtered, not recirculated. Windows are sealed shut. And the lab itself has to be located away from general building traffic, maybe behind double doors or in a separate zone.
And then BSL-4, risk group 4,
Yeah.
the top level. This for the really dangerous stuff, right? Exotic agents, high risk of aerosol transmission, often life-threatening diseases with no available treatments or vaccines.
Exactly. We're talking Ebola virus, Marburg virus, smallpox virus if it were still around outside containment. These are the agents that, frankly, scare everybody.
So the facility must be like a fortress.
Pretty much. BSL-4 labs are usually in a totally separate building or a completely isolated, sealed-off section of one. Getting in and out is a major process. You have to take a chemical decontamination shower every single time you exit. All your street clothes come off when you enter. You put on dedicated lab clothing, which is decontaminated before it leaves.
And supplies.
Everything going in or out has to pass through a double-door autoclave, a fumigation chamber, or a special airlock system. It's all about total containment, absolute isolation.
Okay, that's a clear picture of biological containment. But labs use tons of chemicals too. Acids, solvents, bases. Once you've contained the bug, you have to deal with the chemicals used to study it. How is that managed?
Right. Chemical safety is another huge area. OSHA governs this too, setting limits on occupational exposure to hazardous chemicals. There are key resources workers use. The ALSH Pocket Guide is a big one. It lists important data for chemicals, and maybe not critical for everyone to memorize the numbers, but knowing about things like the IDLH concentration is important.
IDLH, what's that?
It stands for immediately dangerous to life or health. It's basically the maximum level of a toxic substance you could be exposed to for, say, 30 minutes without suffering irreversible health damage or symptoms that would prevent you from escaping. It's kind of a critical threshold concentration, tells you how quickly things can get really bad.
Wow, okay. That definitely defines the risk. And storage is key, I imagine.
Oh, absolutely critical. You can't just stick chemicals anywhere. They have to be properly labeled and stored separately based on their hazard category. Solvents here, oxidizers over there, poison somewhere else, flammables in special cabinets. You can't store things together that could react dangerously.
Like acids and bases or oxidizers and flammables.
Exactly. And every single chemical must have a manufacturer's material safety data sheet, or MSDS, sometimes called SDS now, safety data sheet. These sheets are vital. They detail the chemical's toxicity, health effects, fire and explosion risks, first aid measures, what protective gear you need. It's like the instruction manual for safe handling.
Okay. Shifting gears slightly, radiation. That's another lab hazard. The sources break down into ionizing and non-ionizing. Ionizing is the one we usually think of, right? Radioactive chemicals can cause serious issues like radiation sickness, damage bone marrow.
Correct. Handling radioactive materials requires specific shielding lead bricks, special containers, and personal dosimeters like film badges to track exposure. It's highly regulated. But non-ionizing radiation is actually a much more common hazard in many labs. Think UV light from germicidal lamps or transilluminators used for gels.
UV is bad for DNA, right?
Yep, causes DNA damage, specifically thymine dimers, which can lead to skin cancer with prolonged exposure. It can also damage your eyes, the cornea, and lens. And then there are lasers. High-power lasers can cause nasty thermal burns on skin and tissue, and specific wavelengths can cause photochemical damage to the retina. Standard safety glasses often aren't enough. You need specific filtering eyewear for lasers.
Good point. Okay, quick hits on two more physical hazards. Heat, cold, and noise.
Sure, thermal hazards are common. Autoclaves get incredibly hot, superheated steam under pressure. You need long-sleeved lab coats and, importantly, thick, dry autoclave gloves. Wet gloves transmit heat. On the other end, you have extreme cold. Ultra-low freezers go down to -80 Celsius, and liquid nitrogen is way colder. Handling those requires special insulated gloves and often face shields or goggles to prevent frostbite or cryogenic burns.
And noise. Seems less obvious.
Yeah, but it can be a sneaky one. Equipment like vacuum pumps, centrifuges, or especially ultrasonicators can generate high noise levels. It might not seem bad short term, but prolonged exposure causes permanent cumulative hearing damage. Simple earplugs or earmuffs are often all that's needed, but they need to be used.
Okay, so we know the hazards. Let's talk about the gear that protects us, starting with fire safety.
Right, fire extinguishers. Key thing is knowing the different types, A, B, C, D, K, for different kinds of fires. A for combustibles like wood paper, B for flammable liquids, C for electrical, D for combustible metals, K for cooking oils, grease. And everyone needs to know the pass method for using one. Pull the pin, aim the nozzle at the base of the fire, squeeze the handle, and sweep the nozzle side to side. Training is essential.
And for everyday chemical safety, fume hoods are vital.
Absolutely. They're the primary way to control exposure to noxious or flammable vapors. They work by drawing air away from the person using the hood and exhausting it safely outside, usually after filtering. You have general purpose ones and specialty hoods for things like radioactive work or handling highly corrosive acids like perchloric acid. Using them correctly, keeping the sash at the right height to maintain airflow, is key.
Then there are autoclaves again, essential for sterilization.
Totally essential. Using superheated steam under high pressure kills most microbes. But as you mentioned, operating them requires care. There's the risk of burns from heat and steam, potential for pressure release if opened too soon, and the biological hazard if sterilization isn't complete.
And you mentioned earlier, sometimes things can survive standard autoclaving.
It's rare, but yes. Standard autoclaving is incredibly effective, but some highly resistant bacterial spores, certain archaea from extreme environments, and especially prions, those misfolded proteins that cause diseases like mad cow or CJD, they can be exceptionally resistant. Disposing of prion-contaminated material often requires much harsher treatments, like extended autoclaving at higher temperatures, strong chemical baths, or even incineration. It's a specific, serious challenge.
Okay, so if containment fails or there's a spill, emergency equipment like eye washes and safety showers.
Critical. They need to be accessible really quickly. The standard is usually within 10 seconds travel time from where hazardous materials are used. You have the main plumbed-in showers and eye wash stations. And then there are those smaller, portable, personal eye wash bottles. It's important to remember those little bottles are just for immediate initial flushing to rinse the chemical away while you're getting to the main eye wash or shower for the full required flush time, which is often 15 minutes.
Got it. And the last line of defense, personal protective equipment, PPE, gear worn by the individual.
Right. PPE is what protects you when engineering controls or work practices aren't enough on their own or during specific hazardous tasks. The basics. Gloves are essential for protecting hands from chemicals or infectious agents. Because of increasing latex allergies, non-latex options like vinyl or nitrile are very common now.
Clothing.
Lab coats are standard. They need to be worn fully buttoned or snapped closed, not flapping open. And crucially, lab coats should never be worn outside the lab area. You don't want to carry contamination out. Closed-toe shoes are also a must. No sandals or absorbent canvas shoes.
Eye protection.
Mandatory in almost all lab settings. Basic safety glasses with side shields are usually the minimum, but if there's any risk of splashes or sprays, you need more. Chemical splash goggles, or even a full transparent face shield worn over safety glasses.
and sometimes respiratory protection.
Yes, depending on the hazard. This can range from a simple surgical mask, mainly to protect the environment from the wear or offer minimal splash protection, up to N95 respirators for airborne particles, or even full-face respirators with specific filter cartridges for chemical vapors, or completely self-contained breathing apparatus, SCBA, for highly toxic atmospheres or oxygen deficiency. The level depends entirely on the specific risk assessment.
Okay, let's shift the scene a bit. From the controlled lab environment to clinical settings, hospitals, clinics, nursing homes. The risks here seem broader, maybe more unpredictable.
They are different, certainly. You still have biological hazards, especially blood-borne pathogens, but also chemical exposures. Think disinfectants like formaldehyde or glutaraldehyde used for sterilizing equipment. And ergonomic risks are a huge issue in healthcare, lifting, and moving patients. Regulations still apply. Physicians' offices and clinics fall under OSHA rules, particularly the blood-borne pathogen standard. That requires training, exposure control plans, offering Hep B vaccine, etc.
Hospitals must be incredibly complex.
Oh, massively. Hospital safety programs have to cover everything: chemical safety, industrial hygiene, complex hazardous waste disposal involving infectious materials and chemicals, radiation safety, fire safety, and huge disaster preparedness plans. How do you handle infectious patients during an evacuation? How do you manage public access if there's a scare? It's multifaceted.
And nursing homes have their own specific set of challenges.
They really do. Injury and illness rates for workers in nursing homes are among the highest in healthcare, largely due to those ergonomic strains from patient handling. OSHA has even issued specific guidelines just for nursing home ergonomics. Biologically, a major concern in nursing homes and hospitals, too, is the transmission of drug-resistant organisms, especially things like MRSA methicillin-resistant Staphylococcus aureus. It can spread easily, often carried asymptomatically by patients or even healthcare staff, making control very difficult.
And across all these settings, the big three bloodborne pathogens remain a constant concern.
Absolutely. Hepatitis B, hepatitis C, and HIV. Despite vaccines for Hep B and better treatments, they remain the primary focus for bloodborne pathogen control. Meticulous handling of needles and sharps, universal precautions, proper use of PTE, spill cleanup procedures. It's relentless vigilance.
So wrapping this all up for you, our listener trying to absorb these critical standards, what are the key takeaways? We've gone through the 4 biosafety levels, understood the vital roles of OSHA, CDC, and WHO. We've seen the need for specific training on equipment like autoclaves and eye washes, and highlighted that even the simplest protocols like that 20-second hand wash are absolutely non-negotiable.
If we connect this to the bigger picture, I'd say safety isn't just a list of rules you check off. It's really a dynamic, interconnected system. It relies on good engineering controls building safety into the facility. It relies on clear, well-communicated policies and procedures. And maybe most importantly, it relies on strict, consistent adherence by every single individual. Training is part of that. And it's not static. The system has to be constantly reviewed, updated like the lab manuals from places like LBNL mentioned in the source, or evolving BSL designs to meet new threats and learn from past incidents.
Right. And that brings us to our final provocative thought for you today. The need for safety, especially high-level containment, it isn't shrinking. The source material points out that new BSL-4 facilities, the highest level of containment, are being built or have recently come online in places like Galveston, Texas, and at Boston University. This continuous expansion shows that as science probes deeper into infectious agents, as we encounter new or exotic pathogens, the demand for more advanced, more secure, often physically isolated safety measures just keeps growing. It seems the job of preparing for the next threat is never really finished.